Hi everyone. So here we are together again, ending now this genomics module with this Consolidation Week session. I hope you enjoyed what you've learned so far. For this Consolidation Week, what we are going to do is to cover a bit of what you've learned concerning the command line interface and some of the web resources that you've been seeing, and some others that should be very helpful for you at this stage. Even if we didn't cover everything before, but I wanted to show you a very, very useful tool to work on. So what we will be doing is to take a VCF file that I prepared for you. It contains data for chromosome 1, 2, and 3 of an entire human genome. So I shrunk a bit the data set, but we will be shrinking it a bit more in order to extract the information related only to chromosome 1 using the command line interface. This is made for you to practice a bit, and again the command line interface that you've been using. And it's also meant to lower and to decrease the analysis time once we will be using the web resources. So, by the way, this is a file that contains not any human genome. This is the mutations or small variants that have been reported for Craig Venter human genome. So if you remember, when I talked about the Human Genome Project, I also talked about a private project that was held by a private company that was meant to sequence the human genome. And Craig Venter was the guy leading that company at that time. So this guy sequenced his own genome and he made the data available. So what I've been selecting in this file are the small mutations that are related to chromosome 1, 2, and 3. So what we will be doing together is take out the information related to chromosome 1, as I told you. We will be using the command line for that. And then we are going to use the Ensemble website and especially one tool that is called Variant Effect Predictor online, and we will be using it in order to annotate these mutations. And then you also have kind of a practical, and you will be extracting some information related to that, to these mutations. And I will be showing you throughout this example how different the information you retrieve can be if you use the command line interface to interrogate the annotated file, or if you stick to the web resources, just to make you think about the filters that you are using and how different the results can be if you are not using the correct filters. So let's go first work on this file. Okay, so here is my terminal. What I would suggest is that everyone goes to his own working directory. So you could stop this video at any time and any point and generate these working directories and download on it the file I've been giving you. So let me go to that directory. I would recommend that anyone, sorry, I forgot to go. This is a folder that I put on my desktop. So the name is IBT and Consolidation Week 2017, because I will be using that name throughout the practicals. So let me have a look at what I have in it. And indeed, I put the Venter chromosome 123.vcf file that we will be using. So if you remember well, if we want to extract some particular character from a file, there is a very useful command that is called grep. And this is the one we will be using in order to extract information about chromosome 1. So here I do not have to add the apostrophe. It will be working on my laptop. In case you have a problem, just add the apostrophe. It's working both ways on mine. So let me precise the file I would like to work on, and let me redirect it to a new file that I will be calling chromosome 1. Okay, so let me have a look. Oh, by the way, if there are other characters that do start with chromosome 1 or contain other characters containing chromosome 1, then you should be using an option in grep called -w. But here I know that I do only have one type of character called chromosome 1, so that's fine for me. Okay, so let me have a look at the file that has been generated. So here I do know that the file has been generated, it's okay, but I do not know if it worked well, so I need to double check that the file is not empty. So for that I would use the -l option, and indeed this file is not empty. Let me have a look at this vendor file, the chromosome 1, and it's okay. I do have data for chromosome 1, and what I could do is to double check that it is really ending with a chromosome 1, and it is the case. Okay, so now you have generated data that contains only mutants that are related to chromosome 1. Okay, you could still work a bit on this file, but I think we've been seeing a lot on how to deal with the file. You can count the number of lines. You can do it whether using the grep and pipe it to the WC-L option. We could do that. So it's easy. You just have to do chromosome 1 if you wish from the chromosome 1 file, and then you pipe everything using the WC-L option, and you will be seeing the number of lines. So there is another option that is grep -c that you could use. I'm not, yeah, I can show you the details of it. If you wish, and it's giving exactly the same thing. Okay, now what we are going to do, you can still work on it if you wish, but what I want to do is to move now to the online once you've generated the file you want. And the web resources that we will be using is under the Ensemble website. So I gave you the link to it. And this tool is called Variant Effect Predictor. So we are obviously working with the human genome. So you could go to human and then go to the VEP, but we'll be going directly to it. So we're gonna launch this tool. You have many details. You can still have a look on it. So we are okay to keep the human species as selected, and we would like to upload a file containing that VCF information. So I'm gonna upload it. Sorry, it's this one. Okay, and now you have many kind of information. You have identifiers and frequency data, extra option. You still can have a look on it. I do not have the time to detail everything, but once you kind of put your mouse on it, you have detailed information popping out, so you can be having a look. And I would draw your attention on the polyphen option that you could select in order to have the prediction and score. You've been seeing that, and many other options. For now, I'll be using the default options just to show you how it works. As you can see it could take a while to have that analysis done. So what VEP is doing now is annotating the mutations that we gave him. So it is still running. What I just wanted to show you and to tell you is that it could take a while. I'm stopping the video in order to not to use the whole time I have for that video. You will see that it could take a while. What I just would like to say before the analysis is complete is that I do recommend you to go through the different options we've been selecting, because this would determine the output that we had. I just wanted also to show you how these online resources can help you, even if you do not have enough knowledge using the command line interface to perform these kind of annotations by yourself, or if you do not have the right tools that are installed on your computer. So let's have a look in a moment at what this analysis has output. Okay, now the analysis is done. Let's view the results. So it will give you some statistics on the number of variants that has been processed, on the consequences that are found for these. So do not forget that we are dealing with chromosome 1 only, so these are not datasets for the whole genome, and the coding consequences. So we have frameshift variants. So these are particular terms that you could easily find on internet if you want to have a look at the definitions. Okay, let me just move a bit the window so that you see a bit more. Okay, so you have different kind of informations here. You have the variants that have been uploaded, the location of that variance, the allele that is found, the consequence, the impact, the symbol, the gene, the feature type, the biotype, etc., etc. You have many options here according to what you've selected for using your query. Okay, so I just wanted to stress on some important columns that we will be using during the practical. In fact, why it is important to have the name is just because here, using the filters options, you could select for some of these in order to filter them. So if you have a particular gene name, which will be the case during the practical, you have to select for the gene filter, and according to what you need, select the correct option here, so is, and anything you would like to query. If you would like to look for pseudogenes, for example, it's the feature type option that you would need to see. So let's have a look at the biotype, for example. So you would type biotype. Let's have a look at if there are miRNAs. So if you are in the correct column, the options will appear, and we can be filtering this information out. Now you see here the result of that filtering, which shows you the miRNAs. You have other options which are really interesting. You have a navigation, we will be seeing that after, and you have a download option. So what I would suggest you is to remove any filter first. And the first thing I would really recommend you to do is to download the whole file as a VCF, because we will be working on it later. So I just would like to show you by going back to the terminal that I have loaded this file already, and I've been naming it venter_chromosome1_vep.vcf. I would recommend you to use this name when you download the file. So be careful, it is huge. Do not do it if you do not have enough space and work with a colleague. But you have the size in here. But I would like you to keep these names just because we will be using it as it is for the practicals. So, okay, so I hope you will be enjoying the practicals. Go through it. It is detailed information, and you will be seeing with the concluding remarks why I wanted you to work on these kind of files using both the command line and the online resources. So I hope you will enjoy it. And I'll be taking your question again through Vula if you wish. Thank you and enjoy it.